And welcome, and thank you for joining us. Today, our panelists include the presentations, our presenters from our previous presentations, but again, this is the fourth and final presentation in the Missouri Stream Team Academy's CREEP course on an introduction to EPTs, mayflies, stoneflies, and caddisflies. Here we have Randy Sarver from DNR, who presented on Ephemeroptera last week, and that presentation is available on the Stream Team YouTube page. We also have Barry Polton, who provided the Plecoptera or stonefly presentation a few days ago. Myself, Amy Meyer, with the Missouri Department of Conservation, that did the very first presentation on an introduction. And then today we have Dr. David Bowles from Missouri State University, who will be presenting on Trichoptera or caddisflies. So David, take it away.
Thank you, Amy. First, let me just say thank you for attending today, and also thank you for your interest in this entire process, that being citizen volunteers to get eyes on streams and look at them. I think it's an incredibly important thing to do, and I truly appreciate your efforts to that end. And I hope today, to tell you a few things about Trichoptera or caddisflies. I've been studying caddisflies for 40 years now. I remember the very first caddisfly I ever saw was a larva in a class in about 1980. And I never looked back. It was love at first sight. Excuse me. So that's an adult caddisfly you see in the photograph. And of course, you're going to be looking mostly at the larval forms, which we'll get to here momentarily. There it is. So first, what are caddisflies and where do they fit into the grand scheme of insects? Well, if you look at this phylogenetic tree, the caddisflies are on the right side in that the node 8, and they are endopterygotes, the superorder Endopterygota. That means they have complete metamorphosis. They are related to the beetles and Neuroptera and Hymenoptera and all those different insects, but their closest relatives are the sister order, the Lepidoptera. They share a common ancestor, which we loosely call the Amphismenoptera. They, if you look at this photograph of a fossilized specimen, superficially looks like moth-like, caddisfly-like, somewhere in between. It's neither one. And you see the placement also on here of the Ephemeroptera and Plecoptera as general. And some may argue this tree, but it's generally about right. When did the Trichoptera come into origin from the Amphismenoptera? Well, we know the Amphismenoptera were definitely present in the lower Triassic period. It's possible they were present in the upper Permian. But then sometime in the Jurassic, they split and caddisflies became a distinct order and the Lepidoptera became a distinct order. And the rise of the Lepidoptera occurred in the middle to the upper Jurassic and then the Cretaceous, because something else happened during that period that was kind of big. It was the rise of the flowering plants. The relatives of the Lepidoptera took hold of that, and of course their number of species worldwide just exploded at that point. Another important point here to make is the text I got up there, that just boils down, that's the period of the KT extinction, when some 3/4 of the plant and animal species on Earth went extinct for various reasons. I think it's important to note that many insects, including the caddisflies as we know them today, made it through that perilous time. What are the characteristics of the caddisflies? There are about some 14,500 or so species. It grows yearly worldwide. Most of those are in the tropics, and most of those in tropical regions are in Southeast Asia. That's the center of evolution as we know it for the caddisflies. They are holometabolists. In other words, they have complete metamorphosis of egg, 5 larval stages, pupa, and adults. Both the juveniles and the adults have chewing mouthparts, mandibulate mouthparts, and the adults are somewhat moth-like in appearance. As you can see, that's a picture of a Rhyacophilidae adult on top there, and the wings are covered with CD, or these little hair-like structures, hence the order name, or tricho, referring to the hairy wings. The adults range in size from about 2 millimeters, which are the Hydroptilidae or the Microcaddisflies, to nearly 5 centimeters in some of the Phryganeidae and Phryganeidae that are present in Southeast Asia. We have Phryganeids here in the Ozarks, which we'll talk about here momentarily. The larvae are what we call campodeiform, and rather than try to describe that, there's a picture of a Rhyacophilidae larva, and they're kind of sprawling. The legs are, they're somewhat dorsal ventrally flattened for the most part, and that's a campodeiform larva. They have 5 larval instars, which superficially look alike for the most part. One unique thing about the caddisflies is that they spin silk from labial glands. They have a common duct from which the silk emerges, but they have a labial gland on each side of the head. And that is the reason why they are so ecologically diverse, is because they've adapted to using the silk in many different ways. It helps them form their retreats and their cases. And there's also some free living forms that we'll talk about that still use silk. And I'll save that for a moment, but silk is very important to caddisflies and their diversity. If we can summarize this Trichoptera characteristics with those of its sister order, the Lepidoptera, I think it helps us better understand it. So look at the number of species. There are about 14,500 caddisflies, over 180,000 Lepidopterans. The larval type for the Trichoptera is campodeiform, 5 instars, but it's eruciform for Lepidoptera. And what's an eruciform larva? Visualize a caterpillar, and that's an eruciform larva. So they have the very blocky head, somewhat laterally compressed. The mouth parts in Trichoptera are chewing, both larvae and adults. They are chewing in Lepidoptera larvae, but for the most part, Lepidoptera have siphoning mouth parts. Think of the famous, the coil you see under the mouth of a butterfly. That's for the most part. There are some Lepidopterans that have chewing mouth parts, and most notably is a family of micro moths called the Micropterigidae. They superficially resemble a caddisfly in wing structure and everything. I don't know of a single caddisfly worker that at some point in their career in a blacklight sample found one of these things and think, what the heck kind of caddisfly is that? And then you realize that those are scales on the wings, not setae, and it becomes clear it's a Micropterygiid. So again, setae on the wings for caddisflies, scales for butterflies and moths. Both of them spin silk from labial glands. Caddisflies, for the most part, not all, have lateral abdominal gills, but Lepidoptera, for the most part, do not. The only exceptions to that are the aquatic forms like Petrophila that we see common in our Ozark streams here. And you may encounter those when you do some of your work. The habitat for the larvae is primarily aquatic in Trichoptera, but it's essentially terrestrial for most of the Lepidoptera, exclusive of a few handful of aquatic forms. And then it gets down to the coolness factor. Caddisflies, and I'm somewhat biased here, are really, really cool. And butterflies, you've seen one, you've seen them all. And I do that joke for some of my Lepidopterus friends, and they always act excited about it. But we have fun doing those kinds of things. I do like butterflies and moths. So the real reason why we study caddisflies to begin with, of course, is because fly fishermen. And throughout my career, I've had many times been presented ties that have been tied by fly fishermen, and they'll go, does this look like X genus or X species? And I'll always be real honest with them and say, no, it doesn't look anything like that. But I just joke, this is an art form, and some of them get remarkably close. The one, if you see here on the bottom central, that's to mimic a Limnephilidae-Caddisfly, and that's really good. And of course, the Hydropsychidae larvae in the top right also superficially look like it. Fish really aren't that discerning, so if it's close, they're probably in color form right, they're going to feed on it. Of course, in addition to being very important to fly fishermen as ties, it turns out that caddisflies are also very helpful for us assessing stream condition because of their sensitivities. And again, it boils down to the power of silk. And that is why we have such amazing larval diversity in caddisflies. We have the catch net makers and the tube retreat makers you see in the top left and the middle. And then we also have the case makers. All of those use silk in one form or another to make those structures. And then on the lower left, that's a Rhyacophilidae larva. They do not make retreats or cases. They are free living, but they still use silk. And the way they use that silk is they attach a single silken thread. They attach it to their anal claw and proleg, and then they attach it to a rock, and that is used as an anchor line as they move about in fast currents. Should they get dislodged, they can reel themselves back in, which I think is pretty nifty. I just want to show you this phylogenetic tree to give you an idea how we treat caddisflies in the grand scheme of things. And don't worry about the details of that, but the three major suborders of the caddisflies are the Anulopalpia. Those are the net spinners and retreat makers. And then on the bottom, we see the case builders. That's the suborder Integrapalpia. All of them make cases. And then in the middle, we have 5 families that we call the Spicipalpia. 3 of those families are in the Ozarks. Some of them build cases, some of them are free living, and they are what we call paraphyletic. That means they come from, most likely from multiple sources in our evolutionary history. But we now know that most of these Spicipalpians, as we term them, are most closely related to the Integrapalpia for the most part, but not exclusively. Now, this phylogenetic tree was published in 2016 as the most recent one we have for caddisflies. It's molecular, and so it's incredibly accurate. And there's an interesting thing here. In 1938, there was a man named Herb Ross. He's kind of the father of American Trichoptera. Dr. Ross, in his book, Mountain Caddisflies in 1938, published a phylogeny based only on the morphology of what he saw with his eyes under a 1930s microscope. His phylogenetic tree was almost identical to what Carl Schar published et al. In 2016. The one difference between the two trees is that Ross treated the family Psychomyiidae as a subfamily of the Polycentripotidae. And we now know that those are 2 very distinct families. That was the exception. And I think that was just an absolutely amazing effort on his part. So what is the ecology of the Trichoptera? And I make this very generalized because it's all over the board. But caddisflies occur in all types of aquatic systems. From lakes, there's a few species and taxon families that are in lakes. Primarily, they're in streams, freshwater streams, springs, and those sorts of habitats. There are also a few terrestrial species like Manophylax in Appalachia that occurs just basically on damp surfaces of rocks in the mountains. And then we also have a few species in Australia that are marine. They live in tidal pools along the ocean. That's in itself very unique in insects to find a freshwater insect that lives in a totally marine environment. The larvae are the ecologically important stage, and the greatest duration is spent there. Their life cycles can range from univoltine, in other words, once per year, to bivoltine, twice a year, multivoltine, several times a year. And there's a few that are also semivoltine, in other words, they have a 2-year life cycle. In the Ozarks, most of our species, or most of our families in general, are bivoltine to multivoltine. So particularly in the net spinning caddisflies, it's very common here in streams to have 3 generations during the summer. Others, some of the Limnephilidae have very distinct univoltine life histories here. Their feeding habits are also equally very varied. Some of them are predatory, others are shredders of leaves, where they feed on the fungal hyphae on those leaves to get nutrients. Others are, all of the collector-gatherer groups are the filter-feeding caddisflies. We have some that scrape rocks like Helicocyche to get the algae and the diatoms. Others, some of the Limnephilidae and Phryganeidae, they shred fresh vegetable material. In fact, some of them, there's some Limnephilidae occur in England that are considered agricultural pests of commercial watercress operations, and they're a real pain in the rear end for these guys that try to rear it because the caddisflies get in and wipe out their crop. We don't think of caddisflies in that way here, really, do we? Do the adults feed? I've been asked this question a lot, and the answer is yes. Some of the smaller families that don't live very long, but perhaps a few days, may not feed or very limited, but some of the larger ones, like the larger Phryganeidae-Caddisflies here, do feed. And the way we discovered this was back in the early 90s. A beetle worker named Carl Stefan in Oklahoma started setting out malt traps, like the kind of malt we eat. And he would mix it with water, it would ferment, and then he started collecting all the stuff, and he did that for beetles, but he started collecting a large number of this very large Phryganeidae-Caddisfly called Telostomus, and he got both of our species. And so he gave them to me, and we wound up publishing that, and we hypothesized they were coming to that as a food source to eat, and subsequently it appears that is the case. Most caddisfly larvae are what we would call ecologically sensitive, probably a tolerance value of 4 or less. In other words, they're intolerant of disturbance or perturbations. But there are others that are more tolerant, and that's where they become very handy for the EPT metric, because based on those tolerance limits, and most of them are, I think, fairly accurate. We'll speak specifically to that for each family, but they're mostly accurate, and they can tell us a great deal about water condition. And that photograph, by the way, I just realized that's a stream in northern Thailand, but I just thought it was very pretty. So what do adult caddisflies look like? And here's your answer. They are mostly brown to tawny brown to grayish for the most part. They can range in length from the Microcaddisflies, which is the photograph in the middle on the far left. Those are around 2 millimeters or so, all the way up to the larger one in the middle. That's a Telostomus. That's genus Telostomus. It belongs to the family Phryganeidae, and those can be a good 2 inches. So the size range is all over the place. Most of them, for the most part, are around a 1/2 inch or so in length, if you're looking for a visual reference. And again, they're not particularly showy compared to their sisters, the Lepidopterans. There are some exceptions, like the one in the middle on the bottom. That is a species called Leptoceridae-Exquisita. It's in the family Leptoceridae. It's very common in the Ozarks, and that is by far our showiest caddisfly. And this picture doesn't even do it justice. Those bars are usually yellowish gold. It has those black spots. And if you look at the bottom, the venter, it's usually a really bright emerald green in color. And so when you see these things land on a light sheet or at rest, they're very, very pretty. And so the size range again on these ranges from 2 millimeters or so, and the smallest genus of this in North America is Paucicalcaria, and that's one that I happen to describe from the Arkansas Ozarks. How do we tell the adults apart? Well, we use a variety of characters, and we won't focus on those here, but I wanted to make you aware of them. And they involve what we call setal warts. And those are basically thickened areas of the tissue where the setae come off, and they have very unique configurations for each genus and family. And they occur on the head and the thorax. We also use wing venation, and the spines and spurs on the legs, the number of those and their arrangement is very important in separating the different families and genera. But the most important character, easily, are the genitalia. Each species has a very unique genital configuration, and it's what we call the lock and key hypothesis, because their sclerites are sclerotized and immovable or not flexible. For two specimens to mate, the male genitalia must match that of the female. And so species are very distinct along those lines, and that's one of the major ways we tell the species apart. These are the genital arrangements for 3 of the North American species of Neureoclipsis, which is a polycentropotidae. The one on the bottom, Crepuscularis, is the one we have here in the Ozarks. The pupa of caddisflies is what we call exarate. Compare that to obtect in Lepidoptera. And you see the chrysalis of, I think that's a monarch, in the lower portion of the slide there. All the appendages are tightly bound. They're in the chrysalis. There's nothing you can really see inside that chrysalis. Compare that to the caddisfly pupa. The legs are free, the wing pads are free, the mouth parts are free. They do create a silken polysaccharide pupal skin of sorts, like a puparium, and they are in there, but you can see the free, the legs and whatnot through there. If you look at the top photo on the portion on the right side, in the bottom part of that are some dark areas. Those are the larval sclerites that came off the larva. And we can associate species of caddisflies by using this method where we find mature pupae where they are, you can see the genitalia are formed well enough to make a species call. And then you can match that with the larval sclerites and you can figure out what the larva is. And that works fairly well for a lot of different taxa. And I've done it myself. We don't use pupae too much in taxonomy because they look so much alike. It's very hard to distinguish pupa or pupae among different families and even genera and whatnot. Larvae, and that's why you're here today, because we're going to talk a lot about the larvae. Of course, again, silk is the driver here. It drives their ecological separation and uniqueness. We have, you can see just the wide range of body forms here. And I drew you a Rhyacophilidae with its little silken strand attached to the rock, showing that they also use silk. But it's either the use of silk in cases and gives all these different body forms, or catch nets and retreats. And we're going to go through each family, and we're going to talk about some of the common taxa you expect to see here in the Ozarks and some of their distinguishing characteristics. But first, I just want to tell you about case behavior. And I noticed that Amy has on a necklace today. When she comes back on, you'll be able to see her caddisfly case necklace. But I think there was a man named Bill Stark. He's actually a helicopter worker, but back in the 80s, he started making caddisfly jewelry with Limnephilidae cases, and he would just take the pure cases and make earrings and whatnot. But I had a friend, and he was going to Virginia Tech University. At the same time, I was going to the University of Arkansas. We were working on our PhDs. His name's Ben Stout, and that's a photograph of Ben. And Ben worked on the life history of this species, Pycnopsyche gentilis, and you see it just above his photograph here. That's what their case normally looks like. One of the things he did was he wanted to see if they were selective for materials for cases. And so he started pulling out the larvae of their cases and seeing how long it would take them to rebuild and what they picked and stuff. And one day he just had this eureka moment. He goes, well, if they do that, what if I put something else in there? And so he started putting like aquarium gravel and things of that nature. And sure enough, if that's all you give them, they'll make their case out of that. And so his wife, Kathy, who is in the other photograph there, they got the bright idea to start putting pieces of gold and pearls and turquoise, all sorts of unique minerals, substrates, and these caddisflies would then construct a new case out of these. And then when they pupate and emerge, they would leave the case behind. The cases are very sturdy because silk's very rigorous. And then they would fill them with epoxy and they would make jewelry out of them. Kathy is an artist, and she took this as a business opportunity, in which she still to this day goes to meetings and stuff. And some of you may have encountered her. But she sells that as for her living now, making various jewelry. Unfortunately, Ben passed away in 2018. He developed cancer, and a sad guy, but he was a great scientist. So the other photograph here, you see a Limnephilidae caddisfly. That's the genus Limnephilus. This genus is famous for taking the cases of little gastropods like snails and sticking them on it to make its case out of those. I have personally collected these in the wild, and instead of getting an empty case, they accidentally get a live snail. And so when you pick up the caddisflies, and this is really comical when it happens, but you have the snail moving around trying to get loose, but it's stuck to the back of this caddisfly, and it's quite comical to watch when you see it. But case architecture is very important in caddisflies. There have actually been books written by people like Gary Fontaine and others that look at this unique architecture of caddisfly cases. So enough of that. just a novel thing. I thought you might appreciate that. How do we tell caddisfly larvae apart? Via a variety of characters, and we're not going to dwell on this long since we'll kind of hit at some of them going on. But the main things that separate some of the families are this prosternal horn. And if you look in the photograph on the far left in the upper part, there's a prosternal horn that some people call that a neck spur. The foretrochanter, that's actually the proximal part of the trochanter on the leg. The shape of that is often very important. And then we also use the gills on the abdomen, the anal claws and the anal prolegs, the shape and the configuration thereof. And so there's a variety of characters that we use. If you look at the photograph on the right, you'll see also that there says something about hump, a dorsal hump. Most of the taxa that we're going to talk about, most of the families have these, what we call spacing humps that are used in their case. And we'll get back to that momentarily, but that's a very important character for you to remember. I'll say one more thing here is that if you look at the photograph in the lower left, there's some looks like washboard type plates on the bottom of that head. In the family Hydropsychidae, they have these washboard type plates on the bottom of the head that are used for stridulation. And they're larvae, so it's not used for mating, but we think what they do is it's a way of communicating with other larvae. And as a warning sign, they can actually take their leg and they'll stridulate on those. And remember, Barry talked about the noise making in stoneflies. Well, caddisflies also do that, at least in the Hydropsychidae. And so it's an important communication device for the larvae. And it's probably got to do with spacing their nets and, territoriality and things of that nature. So let's talk about families. The Order Trichoptera in Missouri, there are 27 families in all of North America. 17 or 63% of those are in Missouri, which is pretty good. That's a lot. We have about 230 species in Missouri, and we'll go through these families one by one, but the ones in green are those that do not or are not known to occur in Missouri. They are primarily from about central Texas to throughout the Southwest or west of the Continental Divide. So we don't have them here. As I was looking at this map showing the zones in the primary ecoregions in Missouri, that being the Great Plains and the Eastern Temperate Forest, it kind of struck me that and I don't know if this was done on purpose by the USGS, but I think of Missouri as the agricultural portion, that north of the Missouri River, and the Ozarkian, forested portion below the Missouri River. And so maybe those colors are color coded for the tree counties and the corn counties. I'm not sure, but I would say this. Most of our knowledge about Missouri caddisflies comes from the Ozarkian part. Our knowledge about the northern part is much more scattered, and we really need a good distributional survey of what's going on up there. We really don't have as much information as we do down here in the green part. Apparently people like to have the clear streams we have down here preferentially. So let's talk about these by suborder. We're going to start with the mixed box, the Spicapalpia first, and we'll start you out with the family Rhyacophilidae, which are the free-living caddisflies. We have only one genus, that is Rhyacophila. They are, there is no case. Again, they use the silken safety line to anchor themselves. The distinguishing characters include having a dorsal plate or dorsal sclerite on abdominal segment 9. That's the one right before the anal prolegs. And it's fairly large. It's very obvious when you see it. The anal prolegs themselves are very large. They have very prominent anal claws, and they're free. So they can actually move these around, like, grip things on the substrate. And that's going to be important for separating from the next family. So fairly movable, large anal prolegs and that dorsal plate are key characteristics and no case. Some Western species have gills on the abdomen, ours do not. And one other neat thing about these is often when they're preserved, or sometimes when you find them fresh, they will retract the head back into the prothorax. and it covers about half the head, and so it gives them the effect of somewhat wearing a hoodie when you see this. So it's you may notice that when you collect these in your samples. They like clean streams. They have a biotic index of 0. They are predatory insects. And I was on the Buffalo this weekend collecting, and I and a friend, I probably saw maybe 50 or 60 larval Rhyacophila about. So they're pretty common in clean Ozark streams, and they're usually strikingly green in color for the most part. It can be olivaceous green, but it's almost always green here. The next family is the Glossosomatidae. These are the saddle case makers. They do build a case. It's somewhat like a tortoise shell. If you can visualize that, you can see that fairly well in the photographs. They superficially, as larvae, resemble the Rhyacophilidae. The distinction, well, the similarity is that they also have a dorsal sclera on abdominal segment 9 that's fairly large and plate-like. But they're short. Their prolegs are very short, and the anal claws are very small. Compare those to the Rhyacophila, which, remember, they were much larger. That in the presence of the legs. Sometimes in some of the genera, there are rudimentary gills on the abdomen, but they're usually single and they're very small. And just like Rhyacophilidae, the biotic index of these things is 0. For the most part, they like clean water. The 3 genera present here in the Ozarks are Agapetus, which is very common in Ozark streams. Glossosoma, which is restricted to the larger springs like Merrimack and the Current River, things like that. And then Protoptilla. Protoptilla is the smallest of the 3 genera, and it primarily occurs in springs and in surface streams. It typically is in pool habitats or things of that nature, not necessarily in riffles. So you're not as likely to see larval Protoptilla as you are Agapetus for the most part. Oh, the bane of caddisfly workers. The Microcaddisflies are the Hydroptilidae. Unlike those other 2 families we just covered in the Spicapalpia, these are all over the place in their body forms. We have 10 genera here in the Ozarks: Dibusa, Hydroptilla, Ichthytrichia, Maeotrichia, Neotrichia, Ochrotrichia, Orthotrichia, Oxyothyra, Posycalcaria, I described that one, and Stactobiella. Most of our species are in 2 genera, and those are Hydroptilla and Ochrotrichia. And you can see some of the body forms here. The first thing you'll notice is that they're very small, usually less than 5 millimeters as larvae, even smaller. But they're also the largest in species richness. When I was an up-and-coming student, everybody knew that the largest family species-wise was the Limnephilidae. And then some workers, mainly Steve Harris, and then later myself and a colleague, Mike Mathis, began working on these things, and we described a lot of species. And now the Hydroptilidae is by far the largest family diversity-wise in North America, if not the world. They have 3 very distinct thoracic sclerotized segments. So the others, it was pretty much the pronotum was the only thing sclerotized. But in these, you see 3 sclerotized plates on the pronotum, each segment. If you look at the photograph in the upper right, that is a genus called Ichthytrichia, and it looks like it's been run over by a car, and that's not what happened. That's actually how they look. They're very dorsal ventrally flattened and very sprawling, and they live in cracks and crevices, and particularly limestone streams. They're actually fairly common here in the Ozarks. But they don't often show up in benthic samples because you have to know where they're at and kind of have to dig them out. But they do occasionally find themselves in samples. So if you see that, it's not a mistake. That's Ichthytrichia, and congratulations, you found something that most people never see. Hydroptillids also tend to have a fairly swollen abdomen. And that's going to be important for comparing some of the others. Not that size would matter here, but so the abdomen is somewhat swollen, and then the anal prolegs are very reduced, and sometimes it's pretty much just basically a claw sticking on the side of the terminix of the abdomen. So there's no distinct claws. So you can easily separate those from the previous 2 families. The larval case is of varying materials, and let me go back. So the one in the clear case there, that's a genus called Oxyothyra, and they just use silk only. They won't put anything in their case, so the very neat looking little guys. But the cases are very variable. The neat part is only the fifth instar or the final instar builds a case. The first four instars are free living. In fact, the first instar is what we call a triungulon. It doesn't even look like the other instars. They're somewhat crab-like in appearance or spider-like. You know, their legs are long and they're very fast and they're agitated. They move around more quickly. So that's a triungulon larva. It's the only caddisfly family that has a triungulon. And then again, the first four instars are free living. The biotic index of the Hydroptilidae is very variable. Some are tolerant, like some like Orthotrichia, I think, is a 7. Hydroptila is maybe is a 6. But as a family, the biotic index is 4. That's probably about right for most of them, I think. Some of them are zeros, like Paucicalcaria, hyperendemic genus. That's definitely a 0. But most of the ones you're going to see are definitely 4. So let's talk about the suborder Anulopalpia. And these are the ones you're most likely to see in your samples. And the first one is the family Hydropsychidae, or the net-spinning caddisflies. We have 5 genera here in the Ozarks: Humatispsyche, Biplectrona, that's messed up there, Biplectrona, Hydropsychidae, Macrostemum, and Potamoea. There used to be another distinct genus here called Serratopsyche, but that has subsequently been sunk in as a subgenus of Hydropsychidae. You can see these when you collect, particularly from clean streams. They're the ones that have the checkerboard pattern on the top of the head. And you can see the photograph there of one of those. The one on top in green, that's a Hydropsyche. The one in primarily the white one, that's a Humatispsyche. So the ones you're most likely to see in your stream samples are Humatispsyche and the Hydropsyche. The others, Diplectrona, is primarily a spring inhabiting genus, and the other two, Macrostemum and Potamoea, are primarily larger rivers, and you're not as likely to see those, although they are present here. If you look at the line drawing in the left, it's the dorsum of that head. That's the head of Humatispsyche. And I'll just say this is a novel little way if you wanted to separate the genera. If you look on about the midpoint of the frontal clypeus, there's a notch. Every species of Humatispsyche I know from everywhere has that notch. It's a distinguishing feature of the genus, and it's very prominent in our Ozark specimens. So if you want to go from family down to genus to distinguish Humatispsyche from Hydropsyche, that one would do it. Another key characteristic for separating genera is the trochanter, and that's that again, that extension of the trochanter on the base of the leg, and the configuration of this is very important for distinguishing genera. All of the Hydropsychidae have branched filamentous gills, which are oriented ventrally on the abdomen. You can see the nice photograph down there on the bottom. Their abdomen also appears somewhat hairy. They have a variety of coarse and fine setae distributed abundantly on the abdomen. All 3 segments of the thorax are sclerotized. Remember, they were in Hydroptilidae, so that could be a confusion. You might think you might have one or the other, but size alone is usually pretty distinct, not to mention that the anal prolegs of the Hydropsychids are fairly prominent, and again, they are not in the Hydroptilidae. One of the neat things that attract many people to the net spinning caddisflies are their catch nets. And if you can see that Serratopsyche there on top is built what appears to be a volleyball net. And that net orients into the current, and it catches fine potential food items as they come down the channel, and that's how it gets its food. There's different configurations of those, and the mesh sizes vary among genera and species, so they're very distinctive. And sometimes in streams, particularly with Humatispsyche, you'll see a row, or if you find a bedrock substrate and you'll just see all these little catch nets oriented into the current. And sometimes they are so dense at these locations that they'll actually build another row of catch nets on top of that. So you have like a multi-layer of catch nets going on. And that's perhaps where the stridulation comes into importance there. They, as pupae, they take rocks and they'll use their silk to make those rock shelters around themselves, but they don't build stone cases. All of the hydropsychids are filter feeders, hence the catch net. And the family biotic index is a 4. That's probably pretty accurate for all of the hydropsychids, Hydropsyche. For Chromatopsyche, though, I don't think it is. Chromatopsyche as a genus has a biotic index of 6.6, although I would add that we have 2 or 3 species of Chromatopsyche in the Ozarks that inhabit only clear, small streams, headwater streams, and springs of that nature. I think their biotic index is much lower, possibly a 1 or a 2 at most, but for the genus, it's a little higher. So we'll stick with that biotic index of 4 for the family. I think that's pretty accurate. The next family is the Phylogeny, the fingernet caddisflies. We have 2 genera, Chimera and Wormaldia. Both occur in streams fairly commonly in the Ozarks, although Chimera is by far the most abundant, most commonly represented. They have an elongate head, so it appears narrow in profile. They have a T-shaped membranous labrum, and when that's expanded, and you can see it in the bottom left photograph, it's very distinctive. You can't miss that. Both of them have it. Both of the genera have a very distinct black band at the base of the pronotal sclerite. Only the pronotum has a sclerite. The mesonotum and the metanotum are membranous, so there's no sclerite there. When you see these fresh in a sample, and you have a live sample, if you have Chimera, they are very distinctive because that head capsule is a very bright orange, very orangish in appearance, and the body is very yellowish. So they really jump out at you when you see them. If you find a Wormaldia, they are more cream colored in appearance, and the head is still orange, but not quite as prominently so. But I was collecting some Wormaldia this weekend in the Buffalo. They're fairly common in Ozark streams. If you have a Chimera, the frontal clypeus has a very distinct notch, and often it's very asymmetrical, and we can actually use that to some degree to separate the different species here that occur here, although it's not 100% reliable. But that's another one where if you have a hand lens, you can see that notch, and you can, if it doesn't have a notch, it's Wormaldia. If it does have a notch, it's Chimera. So there's some nice characters for you. Their biotic index for this family is a 3, and I think that's very accurate for Phylogeny. Very, very common in riffle samples in the Ozarks, and probably northern Missouri as well. Oh, one of my favorite families is the family Psychomyiidae, the net-tube caddisflies. We have 3 genera, Lipe, Psychomyiia, and Peduniella. Lipe tends to like springs, seeps, backwaters, and things of that nature. Psychomyiia is very common in clean streams around the state. Peduniella is primarily Ozarkian, mostly in Arkansas, and they like systems that are spring-fed and extremely high quality. They're fairly rare. Definitely a rare insect in Missouri is Peduniella. One of the unique features of the Psychomyiids is that trochanter. Theirs is very almost like you think like a meat cleaver or hatchet shaped, and that will distinguish it from the next family we're going to talk about, the Polycentropotidae. But this is their general appearance. Only the pronotum is sclerotized. The mesonotum and metanotum are not sclerotized. They have a very roundish head capsule, but that trochanter is one of the distinguishing features of them. They have very short anal prolegs with fairly small anal claws. And for retreats, they don't build a catch net. What they do is these graze on stones and whatnot, and they build a silken tube-like retreat. It's not unlike what you would see in some chironomids. And some people probably think that's what they are. So these will show up, you get them scraped off in the samples, they come up. But if you just pick up a rock without disturbing it, if they're present, you'll see these tubes running. And so you have to pinch off one end and then chase them down. And that's where you'll find these Psychomyiidae larvae. Their biotic index is 2, and that's, I think, pretty accurate. They're fairly sensitive organisms. So that brings us to the family Polycentripotidae. They are very abundant here in the Ozarks, or Missouri in general. I should make sure I say Missouri in general. We have 5 genera, all well represented: Cernotina, Cyrinellus, Murioclipsis, Nyctiophylax, and Polycentropus, or Polycentropus, as some say. By sampling in riffles, if you sample in riffle habitats, most of what you're going to find are going to be Polycentropus. Nyctiophylax is also present in riffles to a lesser degree. Not Nyctiophylax. Murioclipsis is also present in riffles, but to a lesser degree. Those are the ones that make the trumpet case. The others, Nyctiophylax and Cernotina and Cyrinellus, they often are found on woody debris, coarse woody habitat, and pools and things of that nature. So you're less likely to encounter those unless you're sampling those specific habitats. Recall the hatchet-shaped trochanter in the Psychomyiidae. In Polycentripotidae, it's very pointed and almost finger-like in appearance, as opposed to hatchet-shaped. So that's one way of quickly distinguishing between the two. Otherwise, they superficially look the same. Another key difference is that the anal legs and claws are much more prominent and larger in Polycentripotidae compared to Psychomyiidae. And you see the picture of the tube, that's a Murioclipsis, and these are easy to spot in streams throughout the state. Just find a water where you can see where it's fairly calm, not a really rapidly flowing riffle, but maybe a glide. Around bridges is usually really good for this, but you can look in the water and you'll see these tube structures. And the larva is at the very bottom of the tube, and they collect material, collects on these, and they come up and feed on it and retreat back into those. They have diverse feeding habits as a family, ranging from predatory to grazers and collector-gatherers. Polycentropus, in this photograph here, both the hind legs and the head, they are almost, for the most part, predatory. And I'll give you a quick tip if you want to separate the two common genera, Murioclipsis and Polycentropus. If you look at the dorsum of the anal proleg in Polycentropus, you see there's like a black X. That's exclusive to every Polycentropus larva I've ever seen from anywhere. They all have it without a question. In Murioclipsis, the other one you're going to get in riffles in Missouri, it's not an X, but rather it's a very distinctive H shape. So it's got the crossbar with the 2 uprights. So that's one fast way to separate those larvae. Their biotic index is a 6, and I think for a family, that's probably pretty accurate. The other and last member of the Annulipalpia is the family Dipseudopsidae. These used to be considered Polycentropotids, but they are now separate. They look superficially like a chimera, like an Ephilopotamide in general appearance, having the orangish head and cream-colored body, but there's some key differences. One is that the tarsal claws are not hook-like, they're very spade-like, almost like a spatula. And that's because these are burrowers. They dig these elaborate, deep, sometimes deep burrows into soft substrate and pools, and they live in those burrows. And you can see the photograph down there of 2 tubes, and then a diagram of what they would look like. They have a very rounded head, not elongate, as you would see in a Ephilopotamide. And one of the key features on this one that really pops out if you get one, or lucky enough to get one in a sample, is this ligula. And the ligula is the labium that's got this modified stylet, and it's probably used in feeding or potentially sensory. But there's a very nice picture of it there. And so it's when you see these, you will not miss it. They're very distinct. So again, that's Phylocentropus. It's our only, we have 2 species. That's our only genus. So you're unlikely to see this one in general habitat sampling, but it's possible you will. Oh, let's talk about the Integrapalpia. This is all the case builders. Check my time. Okay. So what are the Integrapalpia? First, spacing humps. Those are universal for the most part. There's always exceptions. And what are they for? Those spacing humps are, by the way, those are absent in the Spicapalpia. That's one distinct feature. They're located on the first abdominal segment. They can be dorsal, or they can be lateral, or they can be dorsal and lateral. And rarely they are absent entirely. They're an adaptation to get water flow through the case so they can oxygenate those gills. What they do is they hydraulically spread out those spacing humps, and then they force water by undulating their abdomen, and it pulls water through the case and over the gills. This is very useful for separating the families. First family is the Brachycentridae, the Humpless case makers. This is the exception first. We have two genera, Brachycentrus and Microcema. They don't have any humps at all. They have a very distinct pronotum, and you see the dorsal photograph there. They have a distinct crevice or a transverse furrow, and it's very noticeable when you find it. We talked about the prosternal horn under the beneath the head. They do not have a prosternal horn. The pronotum and mesonotum are fully sclerotized, but the mesonotum has reduced sclerites. The case can be either made of woody debris, as you see in this photograph, that's Brachycentrus, or in Microcema, it builds a case out of fine sand grains. I want to reference you back to this with another family that looks superficially like it, but the way you distinguish them, or we'll come back to that point too, but again, they lack a prosternal horn, and they have this distinct sulcus or a transverse furrow on the pronotum, okay? And the legs are often elongate because they're case builders, and they stick those legs out. When they do build a wooden case, it's always distinctly four-sided. The round case is in if it's made out of gravel and Microcema. They're collector-gatherers of parasitin, or they shred woody leaves to get the fungal hyphae. They tend to like clean streams, and so we give them a biotic index of 1. In Missouri, Brachycentrus occurs also in northern Missouri. We have a species up there, but it's also most common in the Ozarks. And Microcema occurs only in the spring-fed rivers of the state, like the Current, some in the Jacks, the 11 Point, that's where we get those. Compare that to the family Lepidoptera. These are case builders. They also build either a wooden case or a stone case. If it's a wooden case, it's four-sided. If it's stone, it's round. They have humps on the lateral sides of the body, on the abdomen, but they lack a dorsal hump. And the antennae, interestingly, is situated right beside the eye. And you can see that in both, it's hard to see in the line drawing, but there's a little bump right beside the eye. And then you can see it in the photograph really well. So there's the antennae right by the eye, and they have a very prominent prosternal horn. And you can see this no matter how you rotate it, just flip the head over, you'll see that horn. So the presence of the lateral humps only and the prosternal horn in this case will make it a lepistomatid. And the eye, and the location of the antenna next to the eye. They are shredders and detritivores, and they are very, very intolerant, having a biotic index of 1. These occur statewide in clean habitats. The Phryganeidae, or some people say Phryganeidae, we have 3 genera, Agrypnia, Phryganea, and Telostomus. That's our largest family by far. These larvae are over 2 inches long. They exclusively build wooden cases, and if you note, the cases, instead of the little pieces like you saw in the previous 2 families, these are big chunks that they orient from, from top to bottom, not crosswise. They have a very prominent prosternal horn. They have prominent unbranched abdominal gills, quite long, and the head always has these distinct pigment stripes. So when you see their head sticking out of a case, Those features in combination make them Phryganeids. Only the pronotum is sclerotized. It's a little bit difficult to see here, but it is sclerotized. The mesonotum and metanotum are not sclerotized. They are either shredders or predators, and they have a biotic index of 4. Quick note, the genus Telostomus, which you see in these two wooden cases here, they often occur in ponds where there are salamanders. And the reason they do is because the larvae have very bold sickle-shaped mandibles, and they are one of the few predators of salamander eggs, which are very tough, and because they're mucilaginous, are very resistant to being popped. Well, these Telostomus larvae will take those sickle-shaped mandibles, and that's what they feed on. They prey on salamander eggs. And it's not uncommon to see both of our species in that genus in salamander ponds in the Ozarks. Kind of a novel thing, I think. The Leptoceridae are the longhorn case makers, and they're called longhorn case makers because the adults have very long antennae, and so do the larvae. I'll show you a photograph in a minute. We have 7 genera: Serralia, Leptocerus, Mystacides, Nectopsyche, Ecetus, Cetoides, and Trionoides. It's possible you're going to get any of these in samples in Missouri. They're all widely represented and fairly common. They build a wide variety of cases ranging from plant materials to stone, all sorts of configurations, very diverse and very large number of species in this family. This is a photograph that shows the antennae. It almost looks like a leg sticking up there. You see, it's probably, pushing half the length of the head. So they're very obvious. Even with a hand lens, you can see these things. And their hind legs are often much longer, sometimes twice as long as the front 2 pairs of legs. And they have diverse feeding habits. Name it, and they do it. Some are predators, some are shredders, some are scrapers, whatnot. The Leptocerids lack a prosternal horn. And again, some of these families have it, and one you might confuse this with, particularly a genus called Trionoides in the Leptoceridae. They look superficially like a Phryganeid, but Phryganeids are bigger, and they have a very prominent prosternal horn. Leptocerids do not. The Limnephilidae, again, we used to think this was the largest family diversity-wise in North America. It is not. It's now second to the Hydroptilidae, which is kind of ironic. One of the largest size families and the littlest of those. The genera we have in the Ozarks are Phrynecia, Glyphopsyche, Ironoquia, Pycnopsyche, Platycentrippus, and Pseudostenophylaks. If you're sampling streams in the Ozarks, by and large, the 2 genera you will find are Pycnopsyche, which are very common here, and Ironoquia. Sometimes those will be out in streams. More often they're on side channels or whatnot. The others are in unique habitats. Like Phrynecia tends to like small spring seeps, as does Pseudostenophylaks like small seeps and springs. And then we have a real oddball. Well, Platycentrippus is very rare. It has only been collected a couple times in Missouri, mostly northern Missouri. But we have a species called Glyphopsyche. Glyphopsyche, Missouri, is hyperendemic. It's known only from Merrimack Springs and in Phillips County, and that's the only population of, and there's 2 species in North America. It's the only population in the world of that species is in one spring in Missouri. And it's definitely a rare, and it's a species in peril, if you ask me. Very unique creature. It gives you some of the photographs of the different case configurations among some of the Pycnopsyche and the Ironoquia and so on. These are large caddisflies. They have a very prominent prosternal horn. They have both dorsal and lateral spacing humps. Both the pronotum and the mesonotum are distinctly sclerotized. The mesonotum may have some sclerites, but they're very small, almost dot-like in appearance, not at all large or plate-like. The antennae is situated about halfway between the mandible and the eye. And another thing you can use to distinguish these from some of the other families is the head. you look, it looks like it's spotted. That's because they have the internal musculature attaches to the head, and those are called, well, they're not, it's not a good name, I don't think, but it's called muscle scars. And that's where the muscle bundles attach inside the head. And so it gives them a speckled appearance. And so that's usually very distinct in limnephilids here. And they have very prominent abdominal gills as well. We give them a biotic index of 4, and I think that's fairly accurate for the most part. Some of the rarer ones like Pseudocinophylax and Phryganeidae, that's probably more closer to 1 or 0. But for the family, 4 catches it pretty well for Pycnopsyche. All the Thrumatidae, they don't really have a common name. And some of you have been around for a while may not realize this name change, but we have one genus here. It's Neophylax. There are 2 species in Missouri. They're fairly widely distributed in temporary streams, clean water streams, small tribs, and that sort of thing. You don't find them in larger rivers. And Neophylax was formerly placed in a family called Unoidae. And then before that, it was placed in the family Limnephilidae. Now it's in the family Thrumatidae. They have both dorsal and lateral spacing humps in their case. They have a distinct prosternal horn, albeit it's not hook-like and large like in the other families, but it's a knob. And when you flip them over and you look, but just in front of the front pair of legs, you'll see this little nubby knob sticking out, and that's their prosternal horn. They'll often, at rest, they will pull their head back into the thorax. And so it, again, much like the Rhyacophilidae and Agapetus, it looks like they're wearing a hoodie. And you can see that fairly well on this top photograph here of that head extending, the pronotum shield extending over the head. Their case is very novel. They build a somewhat dorsal ventrally flattened case. And on each side of it, they put what we call ballast stones. And these are to help hold them in place in faster currents and weight them on the bottom. They always have these stones. And you'll often find these cases still attached to the rocks after they pupate and emerge months after that. And you can pry them loose and there won't be anything in them. But the cases are very neat. They are grazers on periphytons, so that's why you find them in streams that are, have a lot of periphytic growth, so you get some sun exposure. Their biotic index is about 1.6. I think that's about right for the genus Neophylax, although I would note for the family level, it's considered a 4, and that's just not accurate at all for what we have in Missouri, because ours are distinctly much more intolerant. Another point about these is the pupation behavior. They have what we call pupation aggregates. In about August, if you go out, sometimes in July, before they pupate, they all congregate, and you'll often find them on a single rock. So you may find, 30 or 40 of these pre-pupae or pupae attached to a single rock, and they all emerge fairly simultaneously. And I suspect that's to help find mates when they do emerge. They're close by. Again, you may see that when you do your sampling. Neophylax. Oh, the Melanidae. You have to love these guys. They're such novel little insects. We have 2 species in the Ozarks of Melana. They have very prominent dorsal and lateral spacing humps. They do not have a prosternal horn. The head has dark bands that are somewhat reminiscent of what we saw in the Phryganeids. But if you do get these and you're confused, remember the Phryganeids had a very prominent prosternal horn with the head stripes. These have prominent head stripes, but no prosternal horn. And then just the general size is a dead giveaway. Their antennae is weird because it's located near the very front edge of the head, near the anterior leading edge of the head capsule, not in a place you would normally expect to find it. And in most caddisflies, usually it's between the eye and that. And then recall the Lepidostomatids had it right near the eye. The hind leg, like some of the Leptocerids, is very long. You might confuse it with a Leptocerid, but other characters will distinguish it. They have forked gills on each abdominal segment, and they have this really strange case. And it's made out of sand grains, and it's not real firm like other caddisfly cases. It's somewhat loose and floppy, and you can pick it, you can actually bend it. So very odd. They live in primarily sand deposits located in small springs and headwater streams. So if you're looking for these, that's where you'd want to look and collect, is in these little sand deposits. Just sift it, and if they're in there, you'll notice it immediately. They have a biotic index for the family of 6. However, the only genus we have is Melana, and they have an index of 4, so a little more intolerant than the family level. Our next to last family, are the Odontoceridae. Our only species is Marilia. Marilia builds a really neat, firmly packed stone case made out of sand grains. They have both dorsal and lateral spacing humps. The antennae are fairly far forward on the head, as you can see here. In this photograph, you can see it in the line drawings fairly easily. The pronotum, the mesonotum are heavily sclerotized. The metanotum, the third one, is somewhat sclerotized. It has some plates, but they're smaller. Now, you possibly can mix this up with Microcema of the family Brachycentridae, but the way you distinguish them is they don't have a prosternal horn, and they also, remember, in the Microcema, in the Brachycentridae, that furrow was in the pronotum. In Odontoceridae, the furrow is in the mesonotum, the middle segment. So that's a quick way to distinguish the two. And these cases are really hard. Think of it, I think that odonto refers to tooth, so it's very hard and very tooth-shaped case. One weird thing about their cases is that as they add on to it, they tend to start picking up larger rocks as they increase in larval, as the larval instars increase in size. So when you find a case, the upper part, or this end is going to be a little bit larger substrate, which is kind of novel. And they are extremely intolerant. Usually find these in clean waters. They have a biotic index of 0. You don't find them in polluted waters at all. They're very common in the Current River, the Buffalo River in Arkansas. They're very common. Spring-fed rivers in general, they're very common. All right, and that's to think about all for Odontoceridae. By the way, we have one recognized species right now, Marilia flexuosa. There is a second one in the Ozarks. It's in Arkansas. It's known from 2 localities, and it's known only from the larva. But the weird thing about this larva, is that it has characters which place it with a group of Odontocerids from Australia. I and some others, Mike Mathis and I, first found this thing in the 80s at a place near Fayetteville, Arkansas, and they were subsequently found in another place, and I have yet to collect the adults. So now that I'm retired, that may be a new task for me. And finally, we will leave you with the family Helicopsychidae. These are the snail case makers. They built a very unique case made out of sand grains, but it spirals somewhat like a snail. In fact, this is pretty funny. There was a guy named Lee in 1834 described a snail that he called Valvata arenifera. Turns out that his snail was actually Helicopsyche. He didn't look at it closely. He didn't notice, excuse me, that his snail had a head. But that's a little humorous thing, I think. If they're in the case, they're very obvious. Nothing else we have even looks remotely close to that case-wise. But if they're out of the case, and sometimes that happens, look at the anal claw. It's very comb-like, very broad and very comb-like in appearance. They have spacing humps that are very distinct. They lack a prosternal horn. All 3 segments of the thorax are sclerotized, although the meso and meta thorax in some specimens is a little more on the lighter side, but you can still, when you rotate them under a microscope, they're pretty obvious. The Helicopsychids are scrapers. They feed exclusively on algae. That's why when you find them usually on a larger rock or stone and cracks or crevices, that's where they feed on the periphyton. And they can tolerate fairly high water temperatures. It's been a guy named Vince Resch found that they could tolerate water temperatures in California up to 93 degrees Fahrenheit, which is pretty daggone hot. And so the biotic index, this is why this is highlighted in yellow. The biotic index for the family is 3. Our only known genus, 2 species in the Ozarks, is Helicopsyche, and that's often listed as a 0. I don't think it's accurate. And the reason I say that is because of Vince Resch's study, but I did a very large study of caddisflies in eastern Texas, and we published these results. It was we had over collections from over 100 sites, and Helicopsyche was present at the majority of those sites. In some places, it was the most common caddisfly in eastern Texas. And many of these sites were very impaired, highly disturbed systems, and you would collect there and they would be chock-a-block with Helicopsyche. And we proposed that some more study needs to be done on this, that index is probably more like a 6. But for now, you can use it as a 3 in your efforts when you class these, but just bear in mind that this could be changing in hopefully a few years as we get more data on this genus. And I think that is all the families, and I will leave you with this slide. And we talked about caddisflies not being very attractive, kind of brown and tawny. This is the genus Nectopsyche that a colleague of mine worked with in Costa Rica in Central America. As you can see, they are slightly more attractive. This is bordering on butterfly land here. Those are really, really, really pretty insects. And with that, I don't have anything else to present to you, and I'll gladly take your questions.
Okay, thank you so much, David. That was excellent. As you might imagine, we do have quite a few questions, and we'll try to see if we can address the majority of these.
Yeah, I apologize for going over a little bit.
That's fine. I think if folks want to stick around, we can go until about 3:30. Because I think we may end up going. Okay, let me kind of go back up towards the top.
Thank you, Jim.
Okay, I have to scroll quite a bit. Bear with me.
Thanks, Rick.
Okay, so there was a question about the silk. Is it pretty much similar in composition across the different families? And is it in any way, some way lead to the different types of cases?
No, the type of silk, and I don't think there's been a specific study on that, but given the lineage, we assume that that was one evolutionary event, and while there may be some variations in it, it's pretty much the same stuff. And that's also true of some of the closely related insects like the Lepidoptera that spin silk. The silk is very, very similar. And, you know, other things, there's some other insect orders that also can spin silk to various degrees. So probably a one-time event.
Do the caddis collect the debris and carry it along as they build their cases?
Some do, yes. Some create an entirely new case as they get larger because they need larger substrates, but others, like in the Marilia, they will just keep dragging their case along and sticking more stuff on it as they go. And so it just depends.
There's another question that kind of leads to if they actually do leave and re-enter their cases as they're in the larval stage.
Yes, they can. They can re-enter a case. Often, though, if you disturb them too much, and like if you pull Pycnopsyche out of its case, they will often choose just to rebuild a new case. But they can go in and out of a case. It can happen.
Do near constant temperatures of Missouri springs, excuse me, let's see. Oh, wait. Oh, do they alter the emergence schedules compared to streams with more variable temperatures?
They sure do. Give you an example. Well, one is in springs, they tend to have a lower caddisfly diversity, although the abundance of those caddisflies in the springs is like several fold higher than you would get in a stream. The other thing is that, I just lost my train of thought. Springs, Well, let me just shift back to Agapetus. So Agapetus is a species that occurs in springs and also in surface streams. In surface streams, they have a annual life cycle, univoltine life cycle, and they routinely come off in April, May, and that's about it. And they have that egg diapause that goes through the summer. They hatch in like late October, early November, and then they grow rapidly, and then they do it again. In springs in Missouri, they will often have multiple generations. And we have a student at Missouri State that's working on this insect right now. He's doing a repeat of my life history study on the species, but he is looking at it in a spring-fed system. And what we find in Arkansas springs is that they basically emerge all year long. They get a brood, they go through the instars, they lack their egg diapause because of that thermal consistency in those springs. So that's a really good question. And this is probably true of other taxa as well. You go out to the Current River, not Current River, but some of the large volume springs like Big and whatnot, and you're going to find emergence of caddisflies year round. Doesn't matter when you go out. If they emerge on a day it's freezing in the atmosphere, then that's a bad day for them, I guess.
Okay. Another question. Can you explain paraphytic growth?
Paraphytic growth? I'm sorry,
I said it wrong.
Yeah, I made assumptions there. I apologize for that. Paraphyton are the microscopic algae, the diatoms and whatnot, that grow as a film on surface substrate in streams. And so you think of a diatom, which make their silicaceous frustules, and these grow in massive colonies, and they're in algal form, and many aquatic insects are adapted to feeding on those.
I suspect this is a fishing related question. Which species of caddis larvae and adults are most commonly found on the upper current river and Merrimack River? And what is the most common color?
Well, the most common color is kind of a tawny brown, mottled brown. Size would be about, half-inch long is the most common if you're looking for size. So that's the color. And so what was the other part of that was color and your
most common kinds that you find in the upper current and upper Merrimack?
Yeah, the most common ones would be, well, there's a lot of species in the upper Merrimack, and also the upper currents got maybe like, 40 species in it. So, but the most common ones during the summer would be Helicocyche, Serratiopsyche, which is the subgenus of Hydropsychidae, and Chematiopsyche. Those are by far the most common ones that you would get. Chematiopsyche, hands down, and get a lot of Leptocerides too that might be flying through the air. So yeah, there's a lot of different taxa out there.
Is it true that when the more common caddisflies emerge, that they spend very little time on the surface or on the film before they fly away?
Yeah, as soon as they get their wings and they tan, they'll fly. An interesting thing about that, so they get out of the water because they're very vulnerable to predation at that point. But so they have to lay eggs, and so they'll go through their dance flight and they'll mate. And after they leave copula, they have different strategies for laying those eggs, and some will dot them on the edge of rocks along the water line. Others will put them on sticks and whatnot. But in Chimera, several species of Chimera will actually re-enter the water. The gravid female, once the eggs are fertilized, she will actually dive back into the water and lay her eggs on a rock, and then she pops back out the surface. And if the fish doesn't get her, that's their strategy for laying eggs. Very odd, I think. And I've witnessed this myself. It's very fun.
We've got kind of a fun behavioral question. Why do caddisfly larvae gang up on cranefly larvae in my educational display trays? Sometimes more than a dozen will completely cover the cranefly.
Yeah, what they're doing is they're looking for substrate. They're not being like, hey, let's beat up that cranefly. I don't make craneflies might deserve it. I don't know, but so shout to the dipterist. But yeah, it's just a substrate. They will grab onto anything they can because they're used to living on stones in the stream. And so when you knock them loose and they don't have that, they want to get back onto something solid and, that they can seek shelter. And if, those big tupula are pretty big. And so I guess, any port in a storm, so they just jump on their back, hang on.
And I actually had a question, and it's kind of related to another one that was in there. They were asking about the differences between the larval and the pupa stage. which of course the pupa is just the developmental stage between the larva and the adult. My question is, how long do they stay in that pupa stage?
About
Typically.
10 days. 10 days, maybe up to 14, depending on the taxon, but that's the general range. In some, I would suspect, and even in the Hydroptilidae, it might be less because they can come off so fast. But yeah, about 10 days probably.
And I learned a new word, triungulin, the long-legged version of the first 4 instars of the Hydroptilids.
It's a really strange thing. So here's another quick story on that. So I worked on a lot of West Texas Hydroptilidae out in the Desert Springs, and there's a couple of taxa out there. That's one weird genus called Trichoptera, and all 5 larval stages have a triungulin appearance. So they never really take on the appearance of some of the other Hydroptilids, that characteristic body shape. They basically stay in that sprawled appearance their entire life, and they live in these habitats that we call a mediculous habitat. So it's you get these desert springs, and it's the water that flows over the surface of those is a fine film, and so there's never really much of a truly deep aquatic habitat they can live on. So they just set in that flattened condition and graze periphyton, to double back on that term.
Great. I think that covers most of the questions. For those who are wanting to view this later, we will have it up on the Missouri Stream Team YouTube page, and that might take me about a day or so to get that up there. All the other previous presentations are also on the Stream Team YouTube page. The best thing to do is just go to YouTube and search for Missouri Stream Team. And
thank you all very much for listening today. I appreciate your time and tuning in.
It was excellent. Thank you all so much.
If you have more questions, feel free to give them to me. I'll take them at any time.
Oh, actually, I think one more popped in.
I mean, if they want to email me, that's fine too.
Okay. Do you just want to say your email real quick?
Yeah, you can give me my email is my name, David Bowles, all one word, at Missouri State, all one word, .edu.
Okay. Great. Thank you so much.
You're welcome. Thanks, everybody. Appreciate it.